Armando Hasurungan, biology and medicine videos. Please make sure to subscribe. Join the forum and group for the latest videos. Please visit Facebook Armando Hasurungan. Now in this video we will look at toll-like receptors, which are important receptors in the innate immune system. They're found in immune cells. For example, let's begin by drawing out a plasma membrane of an immune cell. Now it can be a macrophage, for example. So the plasma membrane, the outer membrane, is made up of a phospholipid bilayer. Here is the intracellular fluid within the cell and extracellular fluid outside the cell. Here, inside the cell, we also have a double lipid membrane that makes up the nucleus. Here I'm only drawing one lipid membrane, but the nucleus has two lipid membranes. Anyways, within the nucleus is where we find our genetic material, which is DNA. DNA contains genes that we can read and essentially transcribe to make RNA and then proteins. It is important to understand this concept because toll-like receptors initiate this process, you can say. Okay, so toll-like receptors are also abbreviated TLR. And they are plasma-bound receptors, so they are always found on the membrane. In humans, there is approximately 10 toll-like receptors. They have recently found a 11th one in the human gene, but I don't think it actually makes anything. But in mice it does. So, as I mentioned, toll-like receptors help initiate and promote the immune response. But how? Well, toll-like receptors essentially recognizes different foreign agents, components, structures, or materials that don't normally belong to our body. So let's have a look at the different things these toll-like receptors recognize and look at the different toll-like receptors themselves. So toll-like receptor 1 and toll-like receptor 2 are found on the outer membrane here, and they will bind to each other to form a dimer when they recognize things such as peptidoglycans of bacteria, lipoteichoic acids of gram-negative bacteria, lipoproteins, as well as zymosins of fungi. So all these different things will activate toll-like receptor 1 and toll-like receptor 2 that will cause toll-like receptor 1 and 2 to form a dimer. And what happens after this, I'll talk about later. But we'll just focus on what these different toll-like receptors recognize first. Toll-like receptor 2 can also form a dimer with toll-like receptor 6 when they recognize a foreign component as well. When they recognize these same foreign components, such as peptidoglycans and lipoproteins. Now, toll-like receptor 4 recognizes lipopolysaccharides of gram-positive bacteria. And when toll-like receptor 4 recognizes this, it will form a dimer with another toll-like receptor 4. Toll-like receptor 5 recognizes flagella. Notice that toll-like receptor 5 does not form a dimer. Now, toll-like receptor 11 gene, as I mentioned, is found in humans, but don't think they're actually functional. In mice, they recognize uropathogenic bacteria that target the urinary system, as well as profilin. Now, profilin is found in grass or wheat, for example. Anyway, then you have toll-like receptor 10, which I am not sure what it does, or the function is not certain, but it is found on the human plasma membrane nonetheless. Now, you might be thinking, what about toll-like receptor 3, 6, 7, and toll-like receptor 9, which I've skipped or missed? Well, these remaining toll-like receptors are found within endosomes. Somehow, these remaining toll-like receptors begin being expressed within endosomes. So, the story is, when viruses, bacteria, or fungi, they contain their own genetic material, such as double-stranded RNA and single-stranded RNA in viruses, as well as CpG DNA in bacteria, for example. Now, CpG DNA essentially is DNA that contains cytosine and guanine dinucleotides that are unmethylated, but you don't have to know that much detail. Just know it's DNA. So these genetic materials can either enter this cell through endocytosis, or they can be released by the pathogen when the pathogen is also endostized. Either way, the genetic material will be within the endosome. Now, some toll-like receptors are found within the endosome. These toll-like receptors don't need to form dimers. Toll-like receptor 3 recognizes and are activated upon contact with double-stranded RNA or viruses. Toll-like receptor 7 and toll-like receptor 8 are activated upon contact with single-stranded RNA or viruses. Toll-like receptor 9 recognizes and are activated upon contact with CpG DNA of bacteria and fungi, for example. Okay, now that we know the toll-like receptors and what they recognize, what happens next? Well, to put it into simple terms, they become activated. The activation of toll-like receptors will initiate a cascade of events that will essentially lead to the activation of certain transcription factors. The main transcription factors that are activated are AP1, IRF, and NFKB. Now we'll go through each of these transcription factors and what they do in this scenario. But what you have to first understand is that this is a very simplistic diagram. In reality, toll-like receptors activate different transcription factors, but they're all really connected in some way. So let's look at AP1. AP1 stands for activated protein 1, and this will essentially enter the nucleus and transcribe and will stimulate or activate certain genes within the DNA to transcribe a specific RNA. Now, this specific RNA will be translated by ribosomes out in the endoplasmic reticulum. This RNA will be translated to form proteins for cell signaling. AP1 will essentially lead to differentiation, proliferation, and even apoptosis. Then you have IRF and NFKB that can also enter the nucleus. IRF stands for interferon regulatory factor that will activate certain genes in the DNA that will make a specific RNA. Now, this specific RNA will be translated by the ribosomes into interferons. Now, interferons are essentially protein molecules that are a defense mechanism for immune cells or other cells likewise. And we'll talk about them a bit more later. Now, NFKB stands for nuclear factor kappa B, and this is probably the most famous transcription factor. It's like associated with many types of diseases. NF-kappa B will activate or stimulate or activate genes in DNA that will transcribe for a specific RNA that will make essentially cytokines. So the specific RNA will be translated by ribosomes to make pro-inflammatory cytokines. Now, these pro-inflammatory cytokines, as the name suggests, everything to do with inflammation. It will promote inflammation. The main inflammatory cytokine is TNF alpha, but also other cytokines, pro-inflammatory cytokines produced are pro-interleukin 1b and pro-interleukin 18. Now, pro-interleukin 1b and pro-interleukin 18 are actually in an inactive form because they have a pro before them. Now, these proteins, they actually have to be activated, and they are activated by a special enzyme within the cytoplasm called caspase 1. So caspase 1 here is responsible for the proteolytic cleavage of pro-interleukin 1b and pro-interleukin 18. And this will lead to the maturation and release of these two cytokines. So what do all these products do? Well, interleukin 1b and interleukin 18, when they're released, they will essentially enhance the immune response substantially. They will activate lymphocytes, and they also have a chemotactic effect. But they also have other functions that I won't really talk about, but those are the main ones. TNF alpha main function is to enhance the immune response, essentially. TNF alpha also induces apoptosis by binding onto receptors of other cells that are infected or whatever, and these TNF alpha will essentially cause that cell to commit suicide. Now let's talk a bit more about interferons. So interferons are produced when there's a viral infection. You see, the cell will produce interferons and will secrete interferons. These interferons will then signal to other cells about that specific virus and tell the cells to up their defense. So it's a very important communication sort of molecule. So I hope you enjoyed this video on an overview of toll-like receptors. The next video we'll look at the inflammasome and how it's related to this actual diagram, but it is a separate video nonetheless. Hope you enjoyed it. Thank you.